researchers and teachers and students and patients all interacting in different parts of the building, because that's all got to happen for progress to be made in conditions like type 1 diabetes. So this is a living model of how things need to work for what we want to have happen, which is the world to be removed of type 1 diabetes. So part of our role is to turn the passion that's in this room into progress as quickly as possible. And we've done that over a number of decades. And we started by creating the field of research that was type 1 diabetes, because it was only in our lifetimes that people realised there was more than one type of diabetes. And people weren't researching that. And then after a couple of decades of what is now substantial investment, there's some really good lessons being learned. We don't want it to be lessons. We don't want papers. We've got to have those lessons tested in people for safety and efficacy, clinical trials. We've got to have them picked up by small biotech companies and proven a bit further downstream. And we've got to have them then picked up by a pharmaceutical company and then down to a regulatory environment where the health system pays for it and a clinician utilises it, and then a patient benefits. Because that's the bit we care about. If that all doesn't work, things aren't happening as fast as they should. And maybe they will happen, but they might be delayed by a year or 5 years. Every day, you realise these every day. Every day is also another 7 people diagnosed. We do care about that time. We don't just want to wait an extra year or 2 or 3. We want it to happen as quickly as possible, particularly when the end game is in mind as well. So we are working now across that pipeline. We work with basic research, such as is done here, clinical research, such as is also done here. We're now working in early stage companies, not because we want to be an equity investor that gets a great return, but because we want good companies to challenge existing incumbents and to provide private money into this journey. Invest other people's cash. We don't mind if they make a return, as long as it's a return that's caused by a patient getting access. We work with regulatory environments and reimbursement systems. In Australia, we had, in the last election, the commitment made for CGM reimbursement for all children. We wanted it for everybody, but we got it to start with for all children. That's a stake in the ground for publicly funded access to diabetes technologies. If you take a top to bottom view of what areas are we working in, I've talked about the artificial pancreas, technology systems that take over the burden of management and a lot of the risk of management. Now, that started with clunky pumps and then chunky CGMs and then rudimentary algorithms and then maybe pumps that shut off after you're too low anyway, and now pumps that shut off predictably, and now the next generation of technologies is starting to be a lot smarter. We're also learning that we can regenerate cells that are struggling to perform and not clinically relevant. It doesn't actually help you because there's such a small percentage of them available to what should be, but your body's still trying because it's really smart. Last year, there were 3 different ways found, new ways, to regenerate cells. Some from precursor cells, so cells that lie in wait, the second line of offence in your pancreas that can become different types of cells. Beta cells that make insulin, alpha cells, and gamma cells that make other hormones. We can encourage those cells to replicate and develop into what we want, which is insulin-producing cells. There's 3 different teams working on different methods.
When we're talking about stem cells, is that these are cells that have the potential ability to turn into any kind of cell. Okay? So they could turn into a muscle cell or they could turn into a fat cell. And of course in this context, we're trying to turn them into insulin-secreting beta cells. Okay? And the reason why I give Doug here a lot of credit, and by the way, he was funded by the JDRF and still is funded by the JDRF, but the reason why I give him a lot of credit is because, as I say, he transformed the scene, and he published a paper in 2014 that really showed the first promise that the stem cells could actually make something that really looked like a pancreatic beta cell. Yeah? So the cells that he produced, they have insulin present inside the cell, and if you stimulate them with glucose, they will release insulin. And really that paper in 2014 is the first paper that showed that. So after there's a number of different sources for stem cells, but what Doug is doing here and what we're doing in the lab is using induced pluripotent stem cells. So the idea then is that you can take samples, and typically when people talk about it, they talk about a skin sample. So you've got skin cells, and you can transform them back and turn them into stem cells. And at that point there, you will make something, and Doug has made something that looks like an insulin-secreting cell. But if you're thinking of this as a therapy, then there is another major hurdle that you've got to overcome. And that is, if you made a really lovely pancreatic beta cell and you put it back into somebody who has type 1 diabetes, then what would happen is that they would kill those cells. because they would just recognize them as being beta cells and they would kill them. So the other part to this therapy is you've got to have something here that enables you to encase the cell or enclose the cell so you can protect them from the immune response that the body will have. Okay? So when we're thinking of this as a therapeutic approach, you can think of it as a kind of double-sided thing. You've got to make the best cells that you can, and then somehow you've got to protect those cells inside the body. And that's exactly what we're doing here. Rena was recruited into the lab about 8 months ago, something like that. And what she has been doing is following the what we're calling the Melton protocol, which I'm going to describe to you, and we're using that then as a springboard to push forward innovation in the stem cell field. And then finally, we're designing these capsules that will be used to be embedded in a patient and protect those cells. So drawing the cells inside this capsule, protect them from the immune attack so that they can work, they can sense the glucose, they can secrete their insulin, and they're not going to be attacked by the immune cells that are outside of them. So, these guys over here, which includes me, we don't do any work, we just stand around chatting as I am doing now. So the actual people who do the work are all represented here, and you've already been introduced to Rena, who is down here, and she works on the stem cell side. And what we've really found as a key point of distinction of how the cells are behaving in an islet is that each of the beta cells makes contact with a blood capillary within that islet. And these little, so this is an individual islet, and these little sausage shapes that you can see through here are the capillaries that are running through the islet. And what we can see is that each one of those individual beta cells makes a point of contact with the capillaries. And that's shown over here. So this is a single beta cell, and these red things are the capillaries here, and they're making points of contact with that capillary bed. And those points of contact turn out to be very special. They regulate insulin secretion, and we think that they're important for the native behavior of the cell, and we think those are going to be important if we're going to make our stem cells behave like normal. We're actually really uncovering new behavior in the beta cell that hasn't been shown before. And this just gives an example of the kind of experiments that we can do, where we can now extract those individual beta cells. So remember I said that when you extract them, then they don't work very well. We can extract them, and then we can put them into an environment that mimics the environment that's present in the islet. And one of the proteins that we found that is important is this protein up here called fibronectin. And when we put the cells onto a fibronectin environment, then they respond very well. And the responses are shown in these little yellow dots here. Each one of those dots represents an individual insulin-containing granule. And we can see that all of those dots, when we put them on fibronectin, are down towards the bottom where the cell is contacting the fibronectin. Whereas when we've got them on other things, and this is polylysine, the cells don't like it, and actually you can see the insulin granules are all away from that region. So this allows us then to test the mechanisms of how the cells are responding, and we're doing that entirely in a lab environment. So largely what Rena has been able to do since she's been in the lab is actually replicate what he's got. And what you can see here is we're taking these HIPS, stands for human-induced pluripotent stem cells. So they're the cells that you're making from the patient. So you can take these cells, they don't do anything, they just look like a blob, and you can persuade them through these various stages, which as I said takes about 30 days, into something that looks a bit like a pancreatic beta cell. So that's what we got to do. And it's really this very complex protocol that Doug takes credit for, because he's the one that designed the protocol. Then what you can do is you can do all sorts of things. This is called a PAC scan, so you can use this to identify what kind of cells you've got there. You can also stain for the cells, and if we just look in this green down here, the green is showing you where you've got the insulin in these cells. So these are the cells that you produce from stem cells. Now I still think that this is a bit miraculous, right? So if I showed you a stem cell, it just looks like any old cell. There's nothing special about them at all, right? And from those cells, you've actually made something that if you showed me this, I would tell you that's a pancreatic beta cell. Now we can look at those cells, and we can stimulate with glucose. So GSIS is the short name that we use for glucose-stimulated insulin secretion. Okay? So on this axis, we've got insulin. And now what you can see is that in these cells, that when we stimulate them with glucose, so this is high glucose over here, this is low glucose over here, we can stimulate insulin secretion from those cells. Right? So this is really exciting, isn't it? So you've got a cell now that produces insulin and it secretes insulin. I think that is super exciting. If you'd asked me this 10 years ago, in fact I remember sitting through a stem cell talk where somebody was trying to turn stem cells into kidney cells. I just thought it was laughable. So I just thought this was just a ridiculous thing to do. To think that you could reproduce the complexity of development stages. So really what we're talking about is the stages of development that a fetus would go through to produce a kidney. I thought that was ridiculous. But since that time, 10 years ago, then we've got to this stage where we can produce something that actually looks quite like a fetus. And the final thing that I just want to talk about is the capsules, because, as I said, if you made a lovely cell, really look like an insulin secreting beta cell, you've still got a problem with the immune response, right? If you put them back into somebody who has type 1 diabetes, they would kill them immediately. Okay, so you've got to protect them. And that's where these other people come in that are helping us with this project, in the design and the implementation of these chambers. But we don't want that. We need to have the capsule so it's in intimate contact with the body, so that it can sense the glucose and it can pump out the insulin. So the nice thing that these physicists are able to do with this treatment that they've developed is that they can covalently bond proteins to the inside and the outside of this tubing. And those proteins, you can alter those proteins and put different proteins on that can provoke a positive body reaction. Not a negative reaction of fibrosis, but a positive reaction where the body basically accepts that capsule. I spoke about fibronectin earlier on as being something that the cells actually like. And so we can coat the inside with the fibronectin. In addition, then, we can coat the outside, and the importance of this is that it's a stable coating. It's not something that's going to come off. It will be stable for many weeks in the body. We can coat the outside with any proteins, and here we've just got an example which I'm going to show you in the next slide, which is interleukin 4. So what we do with these capsules is we bundle them up together. Right? So what we've done is we've done calculations as to how big they need to be to actually treat either a mouse in this case or a human, so they're not that big, these things. We can bundle them up, and the idea is that the beta cells will be inside here. So here's the bundle that's been shown here, this is what Richard has done, and then we can implant them inside an animal. And that's shown over here. Right? So we've got our mice as our test samples. You can see the bundle that's been implanted here, and then we can measure the responses that the mice have to those capsules. Okay? And we can modify that with our treatment. So the particular treatment I'm showing you over here is interleukin 4, which, as it turns out at the moment, is our favorite protein for putting on the outside of the capsule. It induces a positive response from the body. And one of the positive responses that we can measure here is that it activates these, what so-called N2 macrophages, which are positive macrophages. Right? They have a beneficial effect in the body. And that is shown by the yellow staining that you can see here. So these are the individual fibers. So there's 1, 2, 3, 4, 5 fibers here. This is the control, and this is after we treated them with interleukin 4. And you can see there's much more yellow around these fibers. I hope you can see it anyway, which is indicative of this positive response. That means that these fibers are not going to undergo a fibrotic reaction. The body isn't going to encase them with fibrosis. It can be seen through the fur of these animals, right? So we can just put the camera on them and we can see that luminescence. And what you can see in day 0 is that we've got cells in all of these capsules. Okay? So the cells are alive, they're luminescent. As you go through to day 2, you can see there's no luminescence in this one. And in fact, this capsule here has been untreated. So it's had no treatment on the inside or the outside. And the lack of luminescence there means that all the cells probably have died. Okay? If we come right through to this angle here, which is on day 4, then you can see there's luminescence in this thing down here. And that one is the one where we treated the inside with fibronectin and the outside with interleukin 4. So that was the winner in this particular example. But the idea is that this is now a platform for us then be able to explore all sorts of things, right? So now we've got the ability to actually look basically inside the animal, and not only inside the animal, but inside the capsule, and see how the cells are behaving inside that capsule. And so we can alter the design of our capsule, we can alter the proteins that are inside and outside the capsule, and immediately see what effect they have. Not only that, but we can track this over time. So with the same animal, we can knock him out one day and measure that luminescence, and then 6 months later we can come along and then knock out the animal and check for that luminescence. So right now where we are, we're getting new sciences in all the behavior, we're developing protocols that are going to enhance the stem cell maturation to make them to look more like beta cells. And we're designing capsules and treatments around the capsules to protect those cells also inside the body. Take cells from your skin and convert them into induced pluripotent stem cells. The guy that actually did that was Yamanaka. He's a famous Japanese scientist, as I say. He won the Nobel Prize. When did he win it, Rena? 2011? Something like that, somewhere around 2011. And so this isn't me saying 2030, it's him saying that. So he now has, as you might imagine, he won the Nobel Prize, got a huge consortium of working on stem cells in Japan.